Welcome back. We have been discussing on the phosphoglycerides, and this is the second type of phosphoglyceride where choline is the base in the phosphoglyceride. Okay, so here you can see this choline derivative of phosphoric acid. I already mentioned it is this one. Here you have the choline, and it is attached to the phosphoric acid of the phosphoglyceride. Okay, so when we hydrolyze the phosphatidylcholine, we get glycerol as the alcohol group, fatty acids, two fatty acids repeated, then phosphoric acid, and the phosphoric acid being attached to the amino alcohol, that is the choline. Okay. Now, it is otherwise known as lecithin. You can see here, and lecithin, it is the most abundant phospholipids in the cell membranes. Okay, and phospholipids, it is a very important part of the cell membrane structural framework. And you can see that it is a phosphatidylcholine, which is more abundant among the phospholipids. There are plenty other phospholipids, not only the phosphatidylethanolamine or choline, we have phosphatidylserine, we have phosphatidylinositol, and among all these, it is the phosphatidylcholine which is more abundant in the cell membrane formation. Okay, they also exist just like phosphatidylethanolamine. Phosphatidylcholine also exists in zwitterionic form. They have an acidic end and a basic end. Okay, that is, they can have a positive charge and a negative charge. And it is found that the lecithins, they occur in all the living organisms. And how the name has come actually, lecithin. Okay, lecithos means egg yolk. Okay, and the egg yolks are actually rich in lecithins, and hence they have got the phosphatidylcholine got the name lecithin, right? And like cephalin, they also the lecithins are an important constituents of nerves and the brain tissue. They are a highly polar molecule. The lecithins are highly polar molecule, and they orient themselves in a specific fashion so that the polar end is in contact with the hydrophilic or the polar cytoplasm, while the fatty acid, which is hydrophobic in nature, they usually keep away from the aqueous medium. Okay, so they orient themselves in a specific fashion. And I suppose when you have studied the structure of plasma membrane in your cell biology classes, you have always drawn it in this way, isn't it? Right? So you may have written it as polar head, isn't it? This is being polar head, right? And hydrophobic tail. Fine. So this is how you may have drawn your cell membrane structure. This one. Right. So these here you will have the maybe like cytoplasm, and here the extracellular medium. Okay, which is again extracellular matrix or extracellular medium, which is again aqueous in nature. Okay, it contains more than 70% of water. So here also more than 70% it is composed of water. So what happens? It is these two medium are polar in nature. These two media, isn't it? So these are phospholipids, right? And among the phospholipids, it is a lecithin which is more abundant in nature. And you can see this is the polar head region, and this is the tail region. Okay, what is polar head region formed of? Polar head region is formed of the alcohol group, that is a glycerol, plus you can see here. Here you can see, right? The phosphoric acid, glycerol molecule. Okay, and you will have phosphoric acid, it is attached to the amino alcohol. So it adds up to the polar nature. Okay, so that forms the polar nature. While the two fatty acid molecules, it will be extended away from the cytoplasm or the extracellular matrix. So this is how it is going to have a lipid bilayer, right? So you can see it forms a very important component of the cell membrane, and in the cell membrane you can always see a specific orientation or orderly orientation of the phosphatidylcholine with the polar head group towards the cytoplasmic region or the extracellular region, and the fatty acids away from the two aqueous medium. Okay, so this kind of an orientation actually play a significant role in the structural organization of the cell membrane, right? This you are familiar with from your cell biology classes. Okay, so this was about the phosphoglycerides. We saw two kinds of phosphoglycerides. As per your syllabus, you are supposed to learn lecithin and cephalin. Okay. Now passing on to the, this is actually phosphatidylcholine in detail. Okay, so this is the glycerol molecule, and on two of the alcohol group, it is esterified with the fatty acid. It could be the same or it could be the different fatty acids, right? One alcohol, it is bounded with phosphoric acid or phosphate molecule, and on the phosphate molecule we will be having a choline group in the case of phosphatidylcholine. And these three molecules, that is a glycerol, phosphate and choline, together form the polar head group, while the two fatty acids will be extended to the opposite side, forming the hydrophobic end. Okay, so I hope this is clear. Now next one as we have is a sphingolipids. Sphingolipids, they are again, you can see here, instead of glycerol molecule, they have completely different alcohol which is known as sphingosin. Okay, so other things remaining the same, they have an alcohol group. Sphingolipids do have an alcohol group. Fatty acid attached to esterified with the alcohol group, a phosphate molecule, and some other molecule. Okay, so they are conjugated lipids with alcohol group, fatty acid molecule, phosphoric acid, and some other compound. Okay, but only one thing is, unlike the phosphoglycerides or the glycerophosphates, where you have glycerol as an alcohol, in the case of sphingolipids, they have sphingosin as an alcohol molecule. Okay, so sphingosin, it is otherwise referred as sphingenin, and here you can see this is the molecule, and here these two are the points which is getting actually bound to fatty acid and the phosphoric acid respectively. Okay, here you can see NH2, the amine end, it is getting actually bound to the fatty acid molecule. Okay, so you have NHCOR, right? It is here you can see the CH2OH. The OH is getting again esterified with what you call phosphoric acid unit, and on the phosphoric acid unit you can find the choline or ethanolamine. It could be attached to it. Okay, so this is sphingolipids, and they have a polar head group. What is the polar head group formed of? Polar head group is formed of this one. Okay, so this forms the polar head group. Fine, and non-polar tail. You can see this is non-polar in nature, aliphatic, isn't it? This is non-polar in nature. One. Second is the fatty acid. It is also non-polar in nature. That is hydrophobic. So these two forms the hydrophobic part, and this part it forms the hydrophilic part. Okay, hydrophilic is the polar region, which forms the head region. Okay, now it is sphingolipids. It forms the fourth largest group of membrane lipids. That is those lipids which become a part of the cell membrane. Right? We saw phospholipids are one of the most important group, and after that you can see it is the sphingolipids. Fine. Now the carbohydrate moieties of certain sphingolipids define the human blood groups and therefore determine the type of blood that individuals can safely receive in blood transfusion. And this is what we are talking about. Okay. So what happens is the sphingolipids may be attached to carbohydrate moieties, and in such cases it is these carbohydrate moieties that define the blood group. Okay. Sphingolipids, they are composed of one molecule of the long chain amino alcohol, which is referred as a sphingosin or sphingenin, and one molecule of fatty acid, long chain fatty acid, a polar head group that is joined by the glycosidic linkage in some cases and a phosphodiester in others. Okay, so here you can see, right, a sphingenin, a fatty acid molecule, phosphoric acid. Okay, that is what we are here referring to, a polar head group that is joined by a glycosidic linkage in some or phosphodiester in others. Okay, so here what we see here, it could be a carbohydrate moiety or it could be a phosphoric acid. Okay, if it is phosphoric acid, it is phosphodiester bond. If it is carbohydrate moiety, it is glycosidic bond, which we have already studied under carbohydrates, isn't it? So I hope it is clear. So sphingolipid, it could have, this is just one example where choline is phosphoric acid attached with choline forms the polar region. Okay, polar head region. But sphingolipids can also have a carbohydrate attached to this one through glycosidic bond. Okay, it could also have phosphoric acid and ethanolamine attached to this one. Okay, So other things remaining the same, with the change in this one, the property, the functions, etc. will change. Okay. Now this is what the sphingolipid is about. Now when a fatty acid is attached to sphingolene, we get a ceramide. Okay. So if we don't have any other molecule, if we have a CH2OH here and like a fatty acid here, CHNH2, instead we have CHNHCOR. Okay. So we don't have this part. If we don't have this part, instead we have CH2OH here. The whole compound is known as a ceramide. Okay. So ceramide is formed when a fatty acid is attached to sphingolene at the NH2 path. Okay. And this ceramide, it is actually the it is all the sphingolipids are derivatives of the ceramide molecule. Ceramide in itself, it is present in plasma membrane of animal cells and are especially prominent in myelin. I hope you know what is a myelin. Myelin, it is a membranous sheath that surrounds the axons of neurons. You may have studied